Okay, test one of bioinformatics. Can I drive Stuart's computer without breaking anything? We'll see. Thanks for coming. As Alex said, I've been working for actually since 2002 on the Reactome project. It's a pathway database. I'm going to talk about it because that's the particular pathway database that I know really well, and I think it works for the job that it's intended to do, so I'm happy to talk about it. But one thing I should say at the outset is this is what I'm also trying to show you is a way of thinking about large amounts of biological data, a way of systematically associating the proteins that you might discover or infer from DNA sequencing to make up the proteome of a species of interest. In this case, it's going to be human. Associating that set of proteins with their functions described in a way that allows those functions to be tied together to define a kind of reaction space. What possibly can go on in this organism, or in a relatively simplified, abstracted view of this organism. And that in turn gives us a tool with which we can explore data set, in which we can integrate data, perhaps large amounts of data, perhaps in at least partly automated way, hence trying to make sense out of genomic data floods, and also gives us a way of exploring those data and possibly discovering relationships among things that weren't obvious from looking at smaller numbers of proteins and smaller numbers of relationships. All right, what's the problem? Well, here is a diagram of a biological process, a signaling cascade. You've certainly seen dozens of these trying to make sense out of published papers. We may have read a few ourselves or written a few ourselves, but the point is really that we're showing relationships, functional relationships among proteins. This particular signaling cascade actually comes to bear on two transcription factors, YAP and WWTR, which sit in the cytosol, and from the cytosol, by default, are free to progress into the nucleus, where they will form complexes with other transcription factors that are already present in the nucleus and activate the expression of a number of genes that have growth-promoting downstream features. So we can think of both of these as pro-growth regulatory proteins, and by default, that's what they do. They encourage the cell to metabolize and to proliferate. The entire cascade up above it is a whole array of interactions that tend to keep these two proteins from getting into the nucleus. They can be complexed to form, they can bind another protein to form a stable complex that can't enter the nucleus. They can be phosphorylated, which again makes them non-functional. And those two events in turn, the phosphorylation one in particular, is regulated by an entire cascade of events. And the person who made this picture, as people who make easily human-readable pictures to put in discussion sections of papers in Cell, often do, though this one was drawn by hand for us, but has used colors to distinguish various kinds of proteins from one another, has put little icons on those proteins to indicate where they've been phosphorylated or not. Phosphorylation is an important regulatory feature, has indicated proteolytic cleavage rather graphically, and has used arrows to indicate causal connections. Of course, different causal connections are of different forms: complex formation, phosphorylation, proteolytic cleavage. And so there's a tremendous amount of information here, which is typically immediately obvious to the expert biologist who looks at the picture, and typically 90% inscrutable to a computational algorithm that tries to mine the data out. What does each of these arrows mean? How do I know from looking at this arrow and not reading the crib remark written above that it refers to a phosphorylation event? And who's mediating this phosphorylation? Same here. How do I know that this is a different kind of chemical reaction than this one? We know, but how would how would an algorithm know? And that was the question that we actually set out to, or the issue that we tried to set out to work with in setting up Reactome. And we started from the fairly simplifying and arrogant assertion in 2002 that the process of representing reactions, that one could represent all sorts of processes, not simply classical metabolism, as reactions in which input molecules of whatever sort were transformed into output molecules of whatever chemical sort. The input and output molecules, of course, could be proteins or DNA or other RNA or other macromolecules, or small molecules or complexes. And those transformations could, of course, be the classic chemical conversions of intermediary metabolism, but they could also be transport. A molecule in one location in a cell gets moved to a different and biologically distinguishable location in a cell. For example, it might be moved across a lipid bilayer membrane. A molecule might form a complex with another one. And so we're in effect generalizing a bit on what you think of as a reaction when you think of biochemical ones, but behind it, it's a straightforward extension. There's nothing particularly radical about that idea. And these reactions, of course, exactly as for classical metabolism, can be catalyzed in some instances. Transporters also, in effect, can be annotated as though they were catalysts, because this molecule in location 1 is transformed to this molecule in location 2 by a transporter in the same way that it might be phosphorylated by a kinase. And those reactions, of course, can be regulated by other physical entities, where regulation is, of course, a relatively broad and sometimes fuzzy term, but a really useful one. It can span anything from saying that a molecule might associate with a catalyst to increase its activity or decrease its activity, to something much vaguer, like saying we know that this reaction can't occur except in the presence of some particular entity. We don't know what that entity does, but we know that if it's taken away, the reaction stops. If it's put back in, the reaction resumes again. It's needed only in small amounts. It's not consumed in the reaction, so it can't be an input, but yet it's required. We can also, of course, link individual atomic reactions together into sequences. The output of one reaction might be a required input or a required positive regulator of a second reaction. In the cascade we were looking at, the output of one reaction might be a kinase which is required for the phosphorylation of the next player in the cascade. And without that phosphorylation, the next player can't do anything. So one can make causal connections. We can annotate those manually by saying this reaction to occur requires the previous occurrence of this set of reactions. And once this is underway, this final reaction becomes possible. These kinds of linkages, of course, can also be found if we've annotated things in a consistent data model, can be found computationally. Here is a reaction that produces output 2. Give me a list of all the reactions occurring in the same cellular location that have output 2 as an input, or as a positive regulator, or as a catalyst, and we can say all those other reactions are one step downstream in some biological process or other from this one. And if we have, for example, and this is where user data come in, evidence that all of those components are present in a single cell type at a single time, why then we can infer the computer can infer the existence of a biological process. And likewise, of course, we can manually annotate the existence of one where there's experimental data to do it. To make the information as transparent to users and as interchangeable among data resources as possible, we've defined our domain as being precisely the recognition of what's going on in reactions and how those reactions assemble into pathways. We do not annotate proteins. We pick up the canonical forms of those proteins from UniProt, and where a non-canonical form is used, for example, up here, we describe it within our data. We have tools within our data model to describe the derivative of that canonical form of the protein. In the particular case I just looked at, the protein had a piece chopped off it, so its start and end coordinates have changed from the ones given by UniProt. Other proteins were phosphorylated. We can annotate those modifications by taking that UniProt protein and saying that at a particular residue, using PSI mod terminology, a threonine, let's say, has been phosphorylated, or a tyrosine has been phosphorylated, or as a result of a phosphatase activity, the phosphorylated form of the protein is turned into a dephosphorylated one. We can annotate that. And where small molecules are involved, again, we don't try to work out the chemistry of those. We take them from the Kebi ontology, actually, of small molecules, where we are describing locations in cells. We use terms from the Go cell component ontology to describe the behaviors of catalysts. We use terms from the Go molecular function ontology to describe overall processes. We use Go biological process terms. And in all these cases, the idea is that while we have worked very actively with people at Go and Kebi and to a lesser extent UniProt, if we disagree with their annotations, we deal with that by complaining to them, not by trying to make up a homemade term that will substitute. Okay, and what we found in the time that we've been doing this is that, in fact, this model, within the limits of what kinds of things it can capture, is actually not constrained as to the kinds of biology it can capture. We can do signaling cascades. We can do transport processes. We can do DNA replication and repair. We can do protein synthesis. We can do viral infections. So extending it to, we haven't yet found a domain of cellular biology that we can't annotate. In dealing with cell-cell interactions, we get a little bit shakier, and one of the simplifications we imposed from the very beginning was to say, we're not going to annotate particular cell states and cell types. Rather, we're going to try to build a model of a canonical cell, a generic parts list. A user will come to us with the subset of the proteome that is actually expressed in this cell or in this compartment of this cell, and on that basis, queries us with that parts list, and we give the user back the part of reaction space that's actually operable in the user's cell under the user's physiological conditions. We also, I should say, chose at the beginning not to annotate kinetic data, so all assertions that we make are qualitative ones, because in 2002, we simply couldn't find consistent bodies of kinetic data. Lots of very high quality data, but no way that was within our expertise of mapping up this group's view of, let's say, flux through the pentose phosphate pathway with that other equally expert's view of flux through glycolysis to come up with some combined view of how carbon atoms are moving through, in all the variant forms of glucose breakdown. So those things are not there. Our assertions are qualitative ones. Reactions happen or don't happen, and obviously if a reaction is going at what our experts tell us is a physiologically insignificant rate, that translates into don't happen. And when phosphorylation occurs and the reaction goes at an interesting rate, that translates into does happen. Okay, and what have we, over the time we've been doing this? First, just looking at the standard reporting our progress to the keepers of our NIH contract. We are aiming to have 10,000 proteins annotated by, basically 2 years from now. And these lines track our progress in how many UniProt distinct entries have we gotten. About just under 8,000. In a few cases, we've annotated isoforms because they are functionally distinct. Mostly, the data aren't enough to say that they're functionally distinct. Again, at the granularity we annotate, adding those, there's a couple hundred of those. If you tally up all the modified forms of all the proteins we've annotated, where this includes not only post-translational modifications, but also differences in localization. We've done about 17,000 of those. The number of proteins in the human genome is encoded by the human genome is now estimated to be about 20,000, and informal guesses as to how many of those actually have any experimental data at all associated with them says maybe 15,000. So we'll be about 2/3 of the way towards we're about halfway to completeness now, and we'll be 2/3 of the way within about 2 years. A couple of years ago, we started annotating specifically disease processes. And I'll say what we mean by disease in just a moment. But first, just the bragging statistics. We've so far, and again, in our current release, we've annotated 149 proteins. So that's essentially, again, the canonical forms of 149 proteins, which in turn, and we've associated variations in those proteins with 190 different diseases as defined in the disease ontology. The number of variant polypeptide sequences we've annotated is actually a high number here, 875, and then those have been involved in reactions, pathways, and also in the formation of complexes. So it's saying that, just in terms of pure numbers, we can do this. If we take the entirety of these reactions and display them as a single map of reaction space, we get this view, which actually is something that just went live last week. Each domain, as we've defined it, but again, pretty much consistent with the way that Go breaks up the world into high-level biological processes, is a cluster where each node is a pathway, which has child and grandchild and great-grandchild pathways. Each edge is a collection, represents one of these parent-child links. So if this is all of metabolism at the center, then we might, the immediate subnodes might be carbohydrate metabolism, nucleotide metabolism, and so on, and carbohydrate might have children, glucose, fructose, and so forth. Edges can also link processes that are shared between two different ones of these high-level domains. Again, what this disease cluster is and how it's formed, I'll get to that in a minute. But as I say, there it is. And this actually brings back a feature we had much earlier when our data content was much smaller, that allows users to see the entirety of reaction space at one view, so they can also visualize, by overlaying on it, expression sets that are skewed towards, or for that matter, gene disruption lists that are skewed towards particular processes or away from particular other ones. If we start from this high-level view, say, okay, signal transduction is interesting, let's zoom into the representation of exactly the process I started with, Hippo signaling. It's there in the sky. And we can flip from that to an SBGN semi-compliant view of the details of the process. So this is the same diagram that we started with, except now annotated in a way that accounts for the molecular participants systematically. The color scheme and the shapes of the icons tell you what kind of molecule you're looking at, and whether you're looking at a single molecule or a collection of molecules that are functionally indistinguishable for the purposes of our annotation. Here is YAP all by itself, ready to go into the nucleus and have a positive effect on a downstream transcriptional process. Here are all the things going on, all the reactions going on that can distract YAP and keep it from getting there. And as you actually call up this display on our website, you see it embedded in this other stuff. Here is the panel we were just looking at, our pathway visualization panel. Over here is our entire event hierarchy laid out as in the same way as an analytical table of contents. So our top-level pathways, signal transduction, metabolism, and so on are here, and you can unfurl each one to see its immediate children, and you can unfurl each of its children to see the events and sub-events within it. We also have below the diagram a details panel that shows things like a free text summary of what's going on here, literature references, and some tabs that get you some other features of the display that I'll come back to. There are also a variety of tools that can be invoked directly by clicking on icons in the diagram, or in some cases on edges, and some others that can be opened by clicking here. Just how to optimize these for maximum intuitive user comfort is a work in progress. We've got a lot better at this, but we're still working on it. Oh, thank you. One of the tabs that you can choose is structures, which again is one of our, I think, nicer links out. I really don't want to. If a protein is known, or a complex actually, is known to the protein data bank, if you highlight that protein in the pathway diagram and choose the structures tab down here, you will get links to all of the versions of the structure that PDB has. Again, we are they are the definitive resource for this information. We don't try to improve on it. We haven't sorted this list from best to worst, for example. We pass it on. And if there were anyone in our group, or a user for that matter, who had the expertise to raise a question about any of this, we'd pass it on to PDB. Just as if we or anyone looking at our material says that uniprot protein is incorrect, we'd pass that on. Or that Go term doesn't properly distinguish two different processes, or doesn't identify a human cell component the way it needs to be identified, we regularly pass that on. That's an act of collaboration. As I say, again, has the effect that we can express our opinion of this protein, and you can, via the links from here to Uniprot or PDB or other protein repositories, get to their views of the functions of the same protein, and often in that case, from those resources, often to other pathway databases. So that you can, the links are there to integrate these data across as many resources as are reasonably reliable and as we've thought to make cross-links to. Okay, how do we deal with disease? Well, clearly what we're annotating so far in the normal world is the molecular details of processes. Molecules exist, molecules interact, stuff happens. Okay, so disease. Abnormal molecules exist, abnormal molecules interact in abnormal ways or fail to interact, and abnormal stuff happens, or nothing happens, which is itself an abnormality. So that's what we can annotate without changing anything in the data model, except adding some descriptive tags to the proteins and events to say, hey, this is an abnormal one, not to be confused with its normal counterparts. So what a mutated gene can encode an inactive product. Common, at least in the classic Mendelian disease in humans. A mutated gene encodes a product with a novel function. Classic, common in many somatic mutations giving rise to cancer, where the novel function is often a change from a regulated function to a constitutively active function, or in the case of a tumor suppressor gene, inactivation. Or infection introduces entirely novel genes or gene products that in turn carry out novel functions that disrupt something that's going on that would be going on normally. And so at this level, notice also that disease processes become variants. Each disease process that we annotate, except for infection, which is its own category, but every other disease process that we annotate becomes a variant on a normal one. And so the hierarchy of events and their relationships that we constructed for normal biology becomes a kind of a parody hierarchy of perturbed normal biology. disease phenotypes, we annotate those using terms from the disease ontology and more recently the evidence factor ontology, something that's been developed at EBI as a generalization on disease ontology. And that has the effect right now that someone who comes to us and says, I don't actually care about neurotransmission or apoptosis or all that other stuff. I want to know, tell me what you know about Parkinson's disease. Could, by asking for all the terms that we've tagged, all the reactions, all the proteins that we've tagged with Parkinson's disease terms, could pull out the ones that are sitting in some part of some signaling cascade, the ones that I think we haven't annotated yet that are sitting somewhere related to apoptosis and related to other processes that, because of the way we arrange the molecular event hierarchy, are scattered. It's also possible, because our hierarchy is a directed acyclic graph, not a strict hierarchy, for a user to build a parallel hierarchy that is organized by disease rather than by molecular process. We haven't done it. Someone who wanted to could dump the entire database and content and build such a thing. It's an interesting idea, but right now, where it's actually still under discussion whether using disease phenotypes is enough to allow people to fish out and reintegrate data, or whether we should do it more explicitly. Work in progress. And because of the way we've organized it so far, that diseases are parodies of normal processes, that means we can actually show that explicitly in our pathway diagrams. Here is, we haven't annotated any disease processes relevant to Hippo. There certainly are some, but we haven't done it yet. Here is notch signaling, recently worked through by Maria Milasek. Here is the normal process. Mostly what we're looking at is protein-protein interactions to form complexes. And then as you go off the screen, you see these complexes actually doing something. But what she's annotated here is that mutations in the participating proteins make them incompetent to form complexes. And so instead of having some process that can go off the screen and do something and transmit the signal further, the process halts here. And I put this picture in mostly to show you exactly how disease processes are being shown as straight variants of their normal counterparts, which is actually a very powerful way of integrating and organizing the data when it's biologically legitimate. Okay, so here is a tool for essentially building an archive. What I've shown you so far is what the Alberts textbook would look like if hundreds of people were working continuously to keep it, or parts of it that we've worked on, to keep it actually up to date. And every time you came upon a reference to some other resource that you thought that was interesting, there was a little gnome to go off to the relevant library and pull you out the paper and bring it back to your desk so you could keep on reading. But so in effect we are acting as a passive, I hope, accurate and extensive, but essentially passive repository for what is already known. Can we do something? Can we build tools that allow data analysis and integration? I like to think of this as, can we be interesting as well as useful? So here is a first stab in that direction. As I said, we annotate the sort of differentiation agnostic, state agnostic, generic textbook cell. So what's actually available in a particular cell? Well, this is a molecular biologist's view of the answer to that question, using canned data that are available from a gene expression repository at EBI. What Antonio Fabregat did here was to take a bunch of tissues, as these are defined by molecular biologists. Notice that on this view of things, not only is liver a homogeneous tissue, but so is brain. But we'll ignore that simplification because it's a very standard one. And what's down. So those are the columns. The rows are all the proteins that got annotated in annotating Hippo signaling. And the color here, which can be converted to a numerical scale, shows what the level of expression is of each of these proteins in each of these tissues. White, I believe, is no data. And so that's interesting to look at. And again, to a human biologist looking at a display, this could actually be quite informative for someone who wants to grab the data and take them off someplace where they can be computed over. The whole data, this expression data set can be dumped into a tab delimited table that has a standard format to it. You can take it away and parse it. At the moment, and probably for a long time, because we don't have any concrete plans to change it, you can't bring your own expression data set to this particular display. But we have other ways of doing that I'll come back to in just a moment. A question that you might ask is, well, gets back to the point that our annotation is manual. It doesn't scale particularly well. Work a curator another hour, and you'll get another protein annotated. You want two proteins, you work 2 hours, and so on. And I'm actually exaggerating how fast the curation process can go. So a tool that Guanming Wu built several years ago now asks, can we collect other proteins that might have something to do with a pathway? And the way he addressed that question was to take all the protein pairwise interaction data he could find of whatever quality, but he noted the quality, and assembled them. So for each human protein, he has a list of that protein and all the other all the other human proteins it interacts with, and he used his notes about, his computationally generated notes about quality to generate an aggregate quality score for the protein, so that we can not only say protein A interacts with protein B, but in the world of protein-protein interactions, this looks like a particularly good one or a particularly questionable one. And then, given that long list of protein-protein pairs, he developed tools that will take a protein that is annotated in Reactome and generate a corona around it of all its interactors that are not annotated in Reactome. So you get a corona. This is actually not a corona of interacting proteins, but shows exactly the corona effect. A straightforward extension of that is to take protein small molecule interactions. Same idea. In this case, we haven't aggregated data from multiple sources. Each source that we've grabbed goes in by itself. In this case, it's the Chembl data set of small molecule protein interactions. And we see the interactors of 2 of the players in the Hippo signaling process. We see actually that each of these has some interactors, some small molecule interactors all to itself, but there are also 4 that interact with both. And if one were a medicinal chemist looking for trying to turn STK3 into a druggable target, these might, as given my naive view of medicinal chemistry, these 6 up here might be potential candidates, and these 4 have already been flagged as having at least one off-target effect. It's a useful way to think about the data. Likewise, the protein-protein interactions, one can again build the corona around any one protein, but one can also build the coronas around several, and where one protein touches more than one known target, that may be of interest. It's, this now, other tools built into this are that by hovering over an edge, you can get some information about the interaction, and by clicking on the icon of the partner, you get taken to the appropriate reference page. In this case, you would get to the page for the Chembl entry that this molecule is. And again, so that's useful for a human who's trying to visualize things and navigate. My glib summary is it's a lot like Amazon. You buy a book, and before they tell you how they're going to ship it, they ask you, well, other customers have been interested in this book, have also been interested in these 6 others. Wouldn't you be interested in one of them? I think we're asking a somewhat less self-serving and maybe more interesting question, but it's the same idea. Once you've got that answer, you can look at the answer, as here. You can look at the details, as here. You can also, once again, dump those details into a standardized tab delimited format that can be taken away and parsed. So, you can actually feed this information into into a computational environment and do something with it. It isn't staring at the screen and taking notes. Likewise, I showed you one can, and I should say that this analysis tool explicitly allows user-supplied data sets. You can grab one of our canned ones, and we've tried to make canned ones out of many of the interesting standardized large collections of protein data and protein small molecule data, but you can bring in your own collection of protein, of entity pairs, feed it to us, and to the extent that we recognize the first entity in one of your pairs, we'll overlay the second one in the appropriate corona around it. Okay. Another way in which you can bring your data to us is that you can bring a gene expression data set. I showed you behavior of a canned data set. What the various bits and pieces show here is in back the form that lets you plug in your data. You say, show how these data overlay on pathways. You get exactly the pathway with each entity colored according to its overexpression level from dark blue, not much, to bright yellow, maximal. We spent a long time discussing this color scheme, and kindness to colorblind males won in the end. Lincoln and Robin Hall are both colorblind, so that was definitely carried some weight. Once again, if you zoom up, if you come into the diagram at a higher level, so here you're actually looking at whole pathways and their causal interconnections. The shading here shows you proteins that you're interested in, for which proteins for which we have annotations, but you supplied no data, and then the ones for which we have annotations and you supplied data ranked from least expressed to most expressed. If you give us a time series, you can actually play that time series as an animation, so the various blobs will change color as the expression levels of your proteins change over the course of your experiment. And once again, all the data can be dumped in a tab-delimited analyzable form, and we try to provide some sense of the likely significance of the result in the sense of asking how skewed is your distribution over reaction space compared to what one might expect by chance alone. So if one pathway has 100 proteins in it, so 1/80th of our total annotated data set, we would expect, if your expression set is randomly distributed, that 1/80th of the proteins you've listed would land in that pathway plus or minus sampling fluctuation. And the standard statistical tests for deviation from sampling fluctuation with conservative corrections for resampling are applied. Those tests are certainly too clumsy and probably too conservative, the same as everyone else's. Trying to come up with something that's better is a work in progress, but it's a work in progress in the whole business. And in all honesty, I don't think we have any local particularly good ideas about how to do that better. If anyone out there has good ideas about how to do this, I'd be happy to. I don't really understand the guts of those statistics, but I'd be happy to walk you as far as I do understand and point you to what comes next and give you the email links to the people who really do understand. Okay, another way to look at data is, and this builds actually on that functional interaction network tool that Guanming built as his first go at displaying data this way. He then added to it by saying, let's take a user data set and look at Reactome, and let's take all genes that that we've identified as functional interactors of those things in the user's supplied data set. And let's use those those two combined sets of proteins together with an edge, and then apply an edge betweenness algorithm to try to organize all of those proteins into networks and subnetworks. This is one built out of 79 genes identified in a screen for genes whose variation in humans affect red blood cell phenotypes. And then those grabbed 55 functional interactors, and they assembled computationally into a series of subnetworks, which are distinguished here by color. If you pull out the largest of the subnetworks, there it is. The genes identified in the red blood cell variant screen are circled. The additional ones brought in, proteins brought in as functional interactors, are in diamonds. Interactions for which there's experimental data are solid arrows. Inferred ones are dotted arrows. The tags on the arrowheads indicate the nature of the interaction where that's known. And if you pull out one of them, you say, okay, I'm interested in why are you saying there's something going on involving histone deacetylase 2 and TAL1? You can choose that bar. You can get here is actually the list of all the evidences in favor of an interaction. And here is, the summed quality score calculated by Guanming's Bayesian analysis scheme. And once again, the data, of course, can be dumped into, so you can take them away and do further analyses and computations with them. Okay, so that's the first, and now we get finally to the third part, which is the shortest, but also I think for this particular talk today, also I think the most interesting one. How do we make Reactome? We're all doing data analysis. We're all taking user-generated data sets and trying to find information in those data sets that wasn't apparent to the person who generated them. Hey, tell me what it is. Why should yeah, I can see that this tumor cell is expressing a bunch of proteins that are expressed at much lower levels in the normal counterpart tissue. Is there any significance to that? I see that this group of metabolites is upregulated in response to that, is overexpressed in response to that stress. Can I make any sense out of that? And of course, there's a variety of tools that are already out there doing it. And a question that I think I would like to discuss a lot more and do something about is to ask, how can we turn the kinds of pathway analysis strategies that I've just shown you, skimmed through, into part of that data integration and analysis environment? How can a user who gets back from you, a list of all the proteins that appeared to be disturbed by a set of mutations that were found in genome sequencing, be taken the next step to ask, are there processes that might be specifically or preferentially perturbed by those mutations? And there are some things that are underway with us. I slurred over this point on purpose. Not every entity that's the current display doesn't really let you see the interactors of a complex. We know they exist, but we haven't figured out how to display them properly yet. We also, a long time ago when our data set was smaller, had tools that let a user say, okay, I want to start here at this point in reaction space and get there. So, okay, I want to start at glucose and get to pyruvate. Show me a path to take me there. And we would compute the simplest path that would get you there from the one place to the other, which is fairly straightforwardly doable. That tool, first, we lost the ability to display the results because we simply had too many, until last week, too much data to show in one screen. So you couldn't trace out a path from one place to another. With the new overview of everything, we may have that view back. It's probably time to revive that tool. And of course, once we revive that tool, then it becomes useful, not only for describing normality, but also useful for asking, okay, so now you've shown me how to get from glucose to pyruvate under sort of default conditions. What happens if this enzyme here is mutated? Is there a workaround? What happens if this metabolite isn't available? Is there a workaround? And so one could imagine, actually, the people making knockout mice actually raised this question. It would be lovely to be able to show mouse reaction space, and each time we make a knockout mouse, show us where it hits, because then it might tell us, it might give the people trying to phenotype that mouse some hints about where to look first. Good question. And that one, trying to bring that analysis and visualization tool back to life is actually reasonably high on the developer's to-do list. And one can imagine also building the various kinds of analyses that I've said into a more nearly automated process, so that one could take a data set. Here are the things that are perturbed by the user's experiment, feed them into Reactome, and get them back now decorate in the same way that you might, feed your newly discovered protein into interpro and get a list of all that protein's domains and the function that is often associated with other proteins that have those domains. Here's a kinase domain. Here's a transmembrane domain. Here's a ubiquitination motif. One could feed in and get the much richer and more multidimensional functional annotation that we can provide, and with the additional sets of links out to reference resources. That's the idea. That's the hand-waving idea behind this sentence automated animation tool and annotation tool. And of course, the more that we can work with practical ontology developers, both to enrich their vocabularies and their vocabularies of nouns, the words that describe things, but also their vocabulary of relationships, the verbs that describe connections between things, and do that in an environment where the ontology is being actively used for annotation, the more powerful we and the data that we annotate and feedback to users become as a starting point for integration with other resources. So these are all things that are on my to-do list to pursue here with other people in this room and with other people at Reactome. I should say that I've been working with the project for a while now, but almost everything you saw is the work of other people who are listed here. A group of, basically we are now divided conceptually among three sites. There's a group of people who live in and around New York who cluster at NYU, a group of people who live in England and live actually near Cambridge and Hingston and work at EBI, and a group of people who work in Toronto, as well as a group of people, telecommuters, scattered into Vietnam, Portland, Oregon, and a few other sites who are paid through OICR, so they get listed in this column. But it's a fairly large-scale interaction now among groups here and at EBI, supported by grants from here and from EBI and Europe. And we're trying to build a community resource that will be as useful as we can make it to as diverse an audience as we can make it. And I'd be happy to take questions now and keep on talking afterwards. Do I kill the recording now or wait